Hi everyone. So today's study tip is to teach another student. Teaching is one of the best ways to test your understanding to see if you really remember it. And I think of this all the time when I'm in class and you ask me questions and I have to really think about how to explain this concept, right? So if you can do that with each other and teach each other, it's a great way to remember the information. Now our next chapter is chapter 12, and we're going to start with this part of the muscular system, part 1. So first we got to think about our anatomy, right? So review your anatomy. And you can take a pause right here and think about this. What are our 3 kinds of muscle? And then describe them as voluntary, involuntary, and striated or non-striated. Take a pause and then come back when you're ready. So we have smooth muscle, that's involuntary and non-striated. Cardiac muscle, that's involuntary and striated. And skeletal muscle, that is voluntary and striated. And this is what we're going to talk the most about because we think about it as producing movement, right? Maintaining posture, supporting organs, and producing heat. So we'll talk a lot about muscle contraction today, especially skeletal muscle. Okay. So remember some of the anatomy of muscles, and this is going to be the sub-organ anatomy, or the tissue level anatomy of this stuff. So you have the periosteum covering the bone here, okay? And then a tendon connecting the muscle to the bone. And muscles are surrounded in this fascia, right? This layer of membrane on the outside of the muscle. And then here's actual muscle underneath. And muscles are made of individual muscle fibers, okay? And so you can see the fiber muscle cell here. You put those together into a muscle fascicle. And then you put a bunch of fascicles together to make the skeletal muscle. Within that, within those muscle cells and the muscle fiber, we've got myofilaments that are going to make up that myofibril, and myofibrils make up your muscle fiber. So review from top to bottom or bottom to top the organization of these muscles, right, and how this works. And then also in this picture, I want you to notice the striations, right? We talked about this is going to be a striated muscle, meaning it looks like it's striped under a microscope, right? And so you can see these lines here that make it look striped. Okay. And then there is sarcolemma surrounding our muscle fibers with sarcoplasm in between. So between each of the myofibrils is the sarcoplasm, and then the whole muscle fiber is surrounded in the sarcolemma. Okay. So sarcolemma means, sarco means flesh, and that's the plasma membrane of your muscle fibers or muscle cells. The sarcoplasm is that cytoplasm that's in between. It has lots of mitochondria, right? And this probably makes sense because we need lots of ATP to power our muscle. Different tissues have lots of different amounts of mitochondria, and muscles are going to be one that has some of the most mitochondria to be able to fuel muscular contraction. Okay. The myofibrils are the proteins that actually do the contracting of muscle. And the sarcoplasmic reticulum is going to be the place that's storing calcium in our muscles. It'll be really important for the actual muscle contraction as we start talking about that. So we have a storage of calcium in our muscles. And then we'll talk about these T-tubules, transverse tubules for electrical communication with outside of the cell. Okay, so we need ways in which to have electrical conductivity from the nervous system to control the muscles that will then contract. Right, so now we're at a point where we're going to talk about this point at which the nervous system attaches to or innervates the muscular system. Okay. So another view of the sarcolemma of your muscle cell. Okay, the sarcoplasm in between. And here is showing the different, more of a up and down visual, but particularly pointing out the different parts of these striations. Okay. So there's these lines in the middle are called Z lines. There's a kind of lighter space here and here that are called the I band. The A band is where the actin and myosin are going to overlap. Okay. And particularly important here, I want to show you the sarcoplasmic reticulum. It kind of forms this netting around the outside of the muscle cells, and then with lots of mitochondria in there too. Okay. Okay. So the I band has only the thin filaments, so it's the lightest appearance. The H band is only thick filaments. So that's the central region of the A band. And the A band is where the thick and thin can overlap at the edges. Okay. So here we see we have thick filaments lined up here from the middle. And the thin filaments are going to be the ones that are attached to the Z disc on either side of this singular unit that's called a sarcomere. So the unit, a singular contractile unit of muscle tissue is called a sarcomere. You put sarcomeres together to make those myofibrils, right, that you'll eventually package together to make muscles. Okay. So when myosin pulls actin during this power stroke in muscular contraction, the I band and the H band of the sarcomere decrease. So here's what it looks like when you have a relaxed muscle. Okay, you've got the actin and myosin on there. We see that our H band is pretty big, right? That spot where we only have the thick fibers. Okay. When it starts to contract, that H band gets smaller. The I band gets smaller too. Okay, so there's more overlap then as the myosin pulls the actin. And at full contraction, that H band completely disappears. So we don't see it anymore because the thin and thick filaments are completely overlapping then during the full contraction. So the filaments don't get shorter. Okay, they slide. So you can see how in here in the picture representation, right? We've got the relaxed muscle here with our big H band here. As there's more overlap between the thick and the thin filaments, we have a full contraction. At full contraction, the filaments are at almost complete overlap with each other. So the filaments themselves are not shortening. This is going to make the full muscle shorten, right? But the individual filaments don't shorten. They overlap as they slide. Okay. So let's talk more about this myosin-actin interaction. Myosin is the filament that has two heads to interact with the actin, and the heads are bent. There's a tail region that stays attached and doesn't actually move. The actin is actually a chain of many actin subunits, and each has an active site for a myosin head. It's held together with tropomyosin. We'll talk about this compound coming up. It'll be pretty important in muscle contraction. And troponin helps keep the tropomyosin on the actin. Okay? So we need to think about tropomyosin and troponin and how they affect actin, and myosin and how these two interact. Okay, here's what it looks like. So our thin filaments are made up of two actin threads, okay? So here is the actin thread, and there's kind of two of them that go around each other in this sort of helix formation, okay? You see troponin on each of our actin filaments and tropomyosin running in threads along here, okay? Then you can note the myosin binding sites on the actin. And here's what we mean by the myosin heads and tails. So thick filaments are made up of myosin. You have this tail and the hinge segment that these don't move. So what moves on the myosin is the head portion, okay? Now look at the difference here. So when myosin is bound to actin, the myosin, the myosin head is attached to the myosin binding site on the actin molecule. And look what we have here. We've got ADP and inorganic phosphate. Okay. So that's when it's attached to the thin filament. When you add ATP on here, ATP binds to the ATP binding site on our myosin head. That makes the myosin head come off of the thin filament and bend. So this hinge piece here, it bends more and comes off of the thin filament. Okay, so this process involves calcium and troponin, okay? Calcium binds to troponin. How it does this is it moves tropomyosin and allows for binding. So let's look at this interaction here. In our relaxed muscle, where myosin and actin are not bound to each other, the tropomyosin is here, the troponin is here. In order to contract a muscle, calcium is needed. Calcium binds to troponin. That moves the tropomyosin to open up then the active sites for those myosin heads to bind to the thin filament. Okay, so calcium is required then for muscle contraction because calcium is needed to bind up troponin, move tropomyosin, and allow the myosin heads to bind to the actin filaments. So You can see a couple of things we need already. We need calcium for muscle contraction. We're also going to need ATP. Okay, so this whole thing is called a sliding filament model because the muscle filaments, the actin and myosin, slide, right, to overlap more as those myosin heads attach to and pull on those actin filaments. So the myosin heads hold ADP and inorganic phosphate and energy. Calcium is released from that sarcoplasmic reticulum. Calcium binds to troponin. The tropomyosin-troponin complex is moved aside. And now our myosin binding sites on the actin are free, and so the myosin heads can bind. Then the myosin heads bind to the actin. And that inorganic phosphate is released from the myosin head. Then we get the power stroke of the myosin head filaments. So it has to bind, and then it has to pull on that actin in order for it to slide. Okay? After it slides, then the ADP is released, and ATP binds to myosin to allow detachment. So this feels weird because it feels like ATP should be powering the muscle contraction, but where ATP is actually required is ATP is required to bind to myosin to allow detachment. Okay? And so this is partially how we get things like rigor mortis and prolonged muscle contraction is if there's no more ATP in order for the myosin heads to come off of the actin filaments, muscles stay contracted. Okay? So we need ATP for release. This ATP then will also allow repositioning of this myosin head. So it will turn into ADP and that phosphate and allow that repositioning of the myosin head. Okay, so helps to go through these in order to know what's happening for muscle contraction. Okay. Here's a picture of it. Okay. Showing that at rest, our cross bridges are not attached to the actin. Okay, so here we are, we're unattached. Then a cross bridge forms, right? The myosin head's going to attach to the actin filament. Here's our inorganic phosphate released, causing conformational change of the myosin. Moves that head, which pulls on that actin filament. That's that power stroke, right? So here's our power stroke of that myosin head on the actin filament, causing them to slide. Then ADP is released. A new ATP is going to bind to the myosin head, allowing it to release from the actin. And eventually that ATP is hydrolyzed, causing our cross bridge to return to its original position. Okay, so what you're going to do now is take a quick break and read the short story about rigor mortis in your textbook. And so our question is that the time of a murder victim's death is commonly estimated by the flexibility of the body. So I want you to explain and write down why this is possible. How can we use muscle contraction to estimate time of death? So come back when you've written that and you read this and you're ready to think about it. Okay. So now we're going to talk a little bit about excitation contraction, the process by which action potentials cause contraction. So we have to get from the brain to skeletal muscle, right? When you want to move a muscle, you have to think about that process, and that is your brain causing muscular contraction. So we got to think about this interaction between excitation and contraction and between the nervous system and the muscular system. So how do we get contractions from action potentials and neurons? All right. So this point of connection between the synaptic terminal and the sarcolemma is called a neuromuscular junction. So it's a synapse, right? Just like we had synapses before where they connect to each other. But in this case, it's neuromuscular because you're not connecting neuron to neuron, you're connecting neuron to muscular tissue. So it's a little bit different. But what happens here then to actually, at that synapse, to actually cause muscle contraction is that you're going to have an action potential that releases acetylcholine. So acetylcholine is released into the synaptic cleft. And instead of binding to another neuron like we saw in neural signaling, now that acetylcholine is binding to receptors on the sarcolemma. Okay. And it's actually rippled for increased surface area. So we get this kind of rippled motor end plate. Okay. That acetylcholine binding to the receptor is going to activate and open sodium channels. And so when sodium channels open, just like in the nervous system, we have an influx of sodium that causes an action potential. So this picture is showing you the somatic motor neuron axon coming down, right? And this with the motor end plate here and this neuromuscular junction connecting the nervous system to the muscular system. Okay. One thing I want to point out here is here's the fold sarcolemma for increased surface area. You've got lots of mitochondria in there, right? And here are synaptic vesicles that are then going to put that acetylcholine into the synapse here and eventually bind to the components of this motor end plate. Okay, so the action potential then in the sarcolemma travels along the sarcolemma to T tubules. Now, we said we had to have calcium for muscle contraction. Where is calcium stored? It's stored in the SR. So, the action potential travels along the sarcolemma to T tubules. It opens on the T tubules voltage-gated calcium channels. The voltage-gated calcium channels are coupled to the SR, specifically to the SR calcium release channels. So when the action potential travels along the sarcolemma to T tubules, the T tubules mobilize calcium. They let calcium come into the cell then. So the SR releases the calcium, comes into the cell, and calcium can then have its interaction with that sliding filament contraction. So again, calcium is required. This is one reason why things like blood calcium balance are so important, right? We have to have an ability of cells, like muscle cells that have sarcolemma in them and have SR in them, to have a source of calcium to maintain it so they can then contract. Okay. All right. So to end the contraction, calcium must be transported back into the SR via calcium ATPase pumps. Okay, so that calcium is recycled, it's used again in the next contraction. Okay, so here's showing you this neuromuscular junction, the excitation contraction coupling. Here is our neuron coming down from the central nervous system, right? And it is synapsing, making a neuromuscular junction with our muscle cell. Okay, so here's sarcolemma along the outside with the receptors for acetylcholine in them. And showing you the acetylcholine receptors here where it binds. 2 is going to be the skeletal muscle voltage-gated sodium channels, right? So acetylcholine binds, causes an action potential that moves along the sarcolemma. Voltage-gated sodium channels are opening because of the voltage that got caused from the receptors here. Sodium comes in, okay, activating then these voltage-gated calcium channels that attach to the SR. The SR then can release calcium into the cell. Calcium can do its job with troponin and tropomyosin and allow the sliding filaments to overlap more and contract. Okay. So there's a latent period of about 2 to 3 milliseconds as the action potential moves across the sarcolemma down the T tubules and calcium is released. And that little latent period is before this contraction phase. So it feels pretty immediate to us, but there is a little bit of a latent period here. And some of this, we start to think about limitations of human physiology, right? So you think about sports, how fast someone can run, how accurately they can dunk a basketball. Some of that is going to be a function of your muscles can only be initiated so fast, right? Okay. So after the latent period, then you have this contraction phase where you actually have the binding of the actin and myosin and those power strokes of the myosin heads. And that's when tension is increased in your muscle. Then there's the relaxation phase where ATP is causing detachment of myosin and now tension decreases again. Okay, so it looks like this. Here's our stimulus. Note the latent period, pretty short, but still exists. Okay. Our contraction phase is here, and our relaxation phase is here. So notice it's a lot longer reaction phase than it is a contraction phase. So that contraction is pretty fast. And again, that can do things like give us power when we jump or run, right? And this maximum tension is developed here at the end of the contraction phase before it starts the relaxation phase. And maintaining muscle tension is also a challenge, right? So we think about certain sports that might require that. That's going to be something else to do too. And we can train some of this in our muscles, right? We can cause stronger tension points and we can increase the time of the contraction phase. But some of it is limitations of physiology. Okay. So incomplete technice does not allow complete relaxation. Okay. And so the tension rises then, but it's not stable. Whereas complete tetanus, fast stimuli keep calcium levels high, tension remains strong and constant. And this allows for complete sustained contraction to do work. Okay, so we need to reach this point of complete tetanus for strong and constant tension. Okay. And so when we look at this in muscles, we can see these incomplete tetanus versus complete. But why you might see a decrease then once you reach complete tetanus is that there is muscle fatigue at some point, right? So you do have a decrease at some point and you can't maintain that tension forever. So fatigue occurs when there's not enough ATP to sustain the muscles or as a buildup of carbon dioxide waste, okay? And so maybe you can do things like increase the amount of ATP available. That's possible. Or clear that CO2 waste faster, because both those things can be reasons that muscle fatigue would occur. Okay, so a couple of different videos for you to watch. Again, nice way to review some of these things is to watch these videos and draw out our sliding filament model so you can think about how this contracts. So make sure you know all the steps of muscular contraction and also relaxation, how we get to that point. Okay, and then in our next lecture, we'll continue with part 2 of this muscle chapter.